Hello viewers, welcome to my channel BioQuest Biochemistry classes. Today I am back with a video on glycogen metabolism. Glycogen metabolism consists of two parts. First part is glycogen synthesis, that is known as glycogenesis, and second part is the catabolism of glycogen, that is glycogenolysis. Now to start with glycogenesis. Like any other pathway, we should know what is glycogenesis. Now, let us see what is glycogenesis. It is the synthesis of glycogen from glucose. It requires UDP and ATP, and it requires a protein glycogenin. This is the structure of glycogen. Glycogen is the major storage carbohydrate in animals corresponding to that of starch in plant. But how does it differ from starch? It is more branched in comparison to starch, and it is a branched homopolymer of glucose. In this linear chain, the hexose sugar units are linked by the 1,4 glycosidic linkage, whereas in the branching they are linked by alpha 1,6 glycosidic linkage. Now, why it is important to store glucose as glycogen? We all know that glucose is osmotically active. So, if the reserved carbohydrate for our cell would be glucose, that would have resulted in osmotic damage to our cells. So, it is stored as glycogen. Now, this highly branched structure of glycogen provides a large number of sites for glycogenesis, permitting the rapid release of glucose 1-phosphate for muscle activity. However, the formation of branch point in glycogen is slower than the addition of glucose units to the linear chain. That means how these glucose units are added in the linear chain are at a faster rate than the formation of branching point. How does it help? Because in clinical practice, the endurance athletes practice carbohydrate loading, exercise to exhaustion. That means when muscle glycogen is largely depleted, the athletes consume a high carbohydrate meal, which results in rapid glycogen synthesis with fewer branch points than normal. Now, coming to the site of glycogenesis. It occurs in the cytosol of liver and muscle and a modest amount in our brain. But there is a difference between the glycogenesis in muscle and liver. The muscle glycogen can never produce free glucose because the muscle lacks the enzyme glucose 6-phosphatase. Pyruvate formed by glycolysis from this glucose 6-phosphate in muscle can undergo transamination to alanine, which is exported from muscle and used for gluconeogenesis in the liver. This is known as the glucose-alanine cycle. If you want to see the details of the cycle, you can check my video on gluconeogenesis, the link of which is provided in the I button, or you can check the description box glycogenesis below. It occurs in well-fed state and it is upregulated by the hormone insulin. What happens in well-fed state? There is increase in the blood glucose, or better to say the plasma glucose concentration rises, and this leads to release of insulin from the beta cells of the islet of Langerhans of pancreas, and what happens? There is an alteration of insulin glucagon ratio, and we can see that insulin is a key regulator. It upregulates the enzyme glycogen synthase, which is the rate limiting enzyme of glycogenesis via dephosphorylated. So we have seen what is glycogenesis, synthesis of glycogen from glucose. Where does it occur? It occurs in the cytosol of liver and muscle with a modest amount in the brain. And when does it occur? It occurs in well-fed state, and insulin serves as the important regulatory hormone that upregulates the key enzyme of this pathway, glycogen synthase. Now, what are the steps of glycogenesis? The glucose molecule, the 6-carbon sugar, will be acted upon by the enzyme hexokinase or glucokinase, and it will get converted to glucose 6-phosphate. Now, this glucose 6-phosphate, we all know it may enter the glycolysis, it may enter the pentose phosphate pathway or the HMP shunt, and now we are seeing that it is entering the glycogen synthesis. This glucose 6-phosphate is isomerized to glucose 1-phosphate, and the enzyme for this conversion is phosphoglucomutase. Now, this glucose 1-phosphate will react with UTP, that is uridine triphosphate, to form the active nucleotide UDP-glucose and pyrophosphate. And the enzyme for this pathway is the UDP-glucose pyrophosphorylated. What happens? Here UDP is converted to ATP, and again ATP is again dephosphorylated to ADP. Now, this UTP will interact with the glucose 1-phosphate to form the nucleotide UDP-glucose, and one molecule of pyrophosphate will be liberated, and this pyrophosphate will again be acted upon by the enzyme inorganic pyrophosphorylase, and it will get converted to 2 molecules of inorganic phosphate. Now, this UDP-glucose may enter the uronic acid pathway. It may enter the uronic acid pathway. This UDP-glucose, now it will interact with a protein that is known as glycogenin. It is a 37 kilodalton protein that is glucosylated on a specific tyrosine residue by this UDP-glucose. Glycogenin catalyzes the transfer of a further 7 residues from UDP-glucose in a 1,4 alpha glycosidic linkage to form a glycogen primer, that is the substrate for glycogen synthase. So, what happens with the tyrosine residue of this glycogenin? The UDP-glucose will interact and UDP molecule will get liberated, and this glucosylation of the glycogenin will continue until and unless 8 residues are added, and it will lead to the formation of a primer that is known as glycogen primer. Now, this glycogen primer is the substrate for the rate limiting enzyme of glycogenesis, that is the glycogen synthase. Now, this glycogen synthase will act on the glycogen primer, and it catalyzes the formation of glycoside bond between C1 of the glucose of UDP-glucose and C4 of a terminal glucose residue of glycogen liberating UDP-glucose, and we will see that a linear polymer of glucose is linked by alpha 1,4 glycosidic linkage. Now what happens? Then the addition of glucose residue to a pre-existing glycogen chain or primer occurs at the non-reducing end or the outer end of the molecule, so that the chain of glycogen becomes elongated by successive alpha 1,4 linkages. When at least 11 residues are formed, then a branching enzyme is acting on this linear polymer. And what happens? This branching enzyme will transfer a part of the alpha 1,4 glycosidic linked polymer, at least 6 residues. See here, there are 6 residues, 1, 2, 3, 4, 5 and 6 glucose residues are linked. Here the branching enzyme will occur and it will shift this part to any part of the linear chain, but here the branching will be formed by alpha 1,6 glycosidic linkage. Now what happens? The branching enzyme will act and the glycogen will form. This glycogen synthase, now this part is trimmed off. Again here further residues will act, or here the further residues will start adding here or here, and the glycogen molecule will grow. So the step of glycogen synthase and branching will be repeated many times and we will get the branched glycogen molecule with glycogenin at its core, with the glycogenin protein at its core. Now let us see the regulation of glycogenesis. This glycogenesis, this molecule, this pathway is regulated by 2 ways, like any other metabolic pathway. First one is the regulation by allosteric regulation and second one is by the covalent modification. Now let us check what are those. Allosteric regulation is by the molecule glucose 6-phosphate and covalent regulation is by phosphorylation and dephosphorylation. Now what we have seen? We have seen that glycogenesis occurs in which state? Yes, it occurs in well-fed state. And what happens in well-fed state? The plasma glucose concentration is increased and this leads to release of insulin from the beta cells of the pancreas. So it will be upregulated by insulin. Next part is insulin acts by dephosphorylation. So thumb rule, it will be upregulated by insulin and insulin acts by dephosphorylation. So the key regulatory enzyme or the pacemaker enzyme of the glycogenesis, that is the glycogen synthase, will be active in which form? Phosphorylated or dephosphorylated? Yes, it will be active in dephosphorylated form. Now if insulin is there, the counter-regulatory hormones glucagon and epinephrine, what it will do? It will regulate the enzyme, resulting to down regulation of the enzyme. That is, it will repress the glycogenesis. So it will inactivate the enzyme in phosphorylated form. Now what happens? The glucagon or the epinephrine, they will bind to the G protein couple receptor. As you can see, 7 helical span G protein couple receptor. This is the glucagon. or the epinephrine, they will bind, and here there will be disintegration of the alpha subunit from the beta gamma subunit. G protein is a trimeric protein. So this will lead to the activation of the G protein coupled receptors, and what will happen? This will lead to the activation of the enzyme adenylyl cyclase or adenylate cyclase. It will lead to the activation of the adenylyl cyclase or the adenylate cyclase. Now, what does this adenylyl cyclase or adenylate cyclase do? It will act upon the ATP, and ATP will get converted to cyclic AMP. Now, this cyclic AMP will activate the enzyme cyclic AMP dependent protein kinase. It will activate the enzyme cyclic AMP dependent protein kinase, and it will be activated. Now, this active protein kinase, what it will do? It will act on the dephosphorylated form of glycogen synthase and inactivate it by phosphorylating it. So, cyclic AMP will activate protein kinase via which the glycogen synthase will be phosphorylated and it will be inactivated. And in well-fed state where we have insulin, what this insulin will do? This insulin will activate the protein phosphatase 1 and it will again dephosphorylate the glycogen synthase, and glycogen synthase will be activated. This is one way where insulin regulates the glycogen synthase activity. Another way, it will inactivate the cyclic AMP. What it will do? This cyclic AMP, if it is acted upon by the enzyme phosphodiesterase, it is converted to 5' AMP and the activity of cyclic AMP is lost. Now, this phosphodiesterase is activated by insulin. So, this is how insulin regulates the activity of the glycogen synthase in our physiological system. This is how it regulates the activity of the glycogen synthase in our system. Now, here there is a common catch question. This inactive protein kinase A will activate to active form protein kinase A, and this cyclic AMP dependent protein kinase, it will in between activate another enzyme that is known as glycogen synthase kinase, that is known as glycogen synthase kinase, that is known as glycogen synthase kinase. So, this is how the regulation of glycogenesis takes place. So, to sum up, glycogenesis is the synthesis of glycogen from glucose, and it requires UTP and ADP and a protein that is known as glycogenin. Where does it occur? It occurs in the cytosol of liver and the muscle, but muscle can never serve as a reservoir for free glucose because muscle lacks the enzyme glucose 6-phosphatase. Next, when does it occur? It occurs when we are in well-fed condition. That means the glucose concentration is very increased in our plasma. It leads to release of insulin from the beta cell of the pancreas. Now, why it is stored as glycogen? Because glucose is osmotically active and that may cause osmotic damage to the cell. Next, what are the steps of glycogen synthesis? First is formation of glucose 6-phosphate. Second, isomerization of glucose 6-phosphate to glucose 1-phosphate. Then formation of UDP-glucose. Now this UDP-glucose will interact with a protein that is known as glycogenin that has a tyrosine residue, and this will lead to autoglucosylation of the glycogenin until and unless 8 residues are added, and this serves as the glycogen primer. Now this glycogen primer will be acted upon by the key regulatory enzyme or the pacemaker enzyme of the glycogen synthesis, glycogen synthase, and it will get converted to a linear polymer until and unless 11 residues are there, that the branching enzyme will act and it will trim 6 residues and they will be added in any point by alpha-1,6 glycosidic linkage. Again, the growth by alpha-1,4 in the non-reducing end will take place in different directions and a highly branched glycogen polymer will be formed with the glycogenin molecule at the center. Now, what are the steps of regulation? Allosteric regulation by glucose 6-phosphate and the key regulatory enzyme glycogen synthase. It is active in dephosphorylated form. Insulin via protein phosphatase 1 and by the degradation of cyclic AMP by phosphodiesterase 8 upregulates the glycogen synthase, whereas the glucagon and epinephrine via G protein coupled receptor via the adenylyl cyclase, it converts ATP to cyclic AMP. Now the cyclic AMP will inactivate protein kinase A that will upregulate glycogen synthase kinase, and glycogen synthase will be phosphorylated and inactivated. So this is how glycogenesis is occurring in our body. These are the details of glycogen metabolism in our system. Glycogen synthesis has appeared in various exams as a LAQ, SAQ, and it is very important. So be prepared for this topic, and if you like the video,